everyone. I'm Glenys Scadding, and it's my great pleasure as President of the UK Semiochemistry Network to invite you to our third virtual meeting this year. It's been organised by Professor Krishna Persaud, and it deals with, I have to read this, synthetic diamond-based sensors for artificial olfaction, from sensor development to field application. And since it contains the word diamonds, I am automatically highly interested. Our first speaker is Professor Carla Mucignat, who is from the University of Padova in Italy. She's interested in the biological effects of major urinary proteins and also in their biotechnical exploration. Over to you, Carla.
Thank you so much, Glenys, for this kind of presentation. And let's start sharing the screen. Okay, so let's start with a little bit of the history about the proteins we are dealing with. And I will start by introducing odorant binding proteins, just by saying that the history of odorant binding proteins started in 1981, where when two different groups described odorant binding proteins, or proteins which are able to bind odorants, in a moth and in the nasal mucus of the cow. And we see connected here at least one of the fathers of these proteins, actually. And just have a look at invertebrate odorant binding proteins. Actually, there are present also other proteins in invertebrates which are able to bind odorants, and these are called chemosensory proteins and were described a little bit later in 1994. All of them are small proteins around 15 kilodaltons, made up of a structure made of alpha helices encompassing a binding sites, stabilized by disulfide bridges, which may be 3 in OBPs or 2 in CSPs. But let's have a closer look to invertebrate OBPs. They are only found in hexapoda, but not in crustacea and myriapoda, which means that they emerged in the last 100 million years, and they constitute a huge family of proteins. Let's say in the fly, they are more than 80, in the silkworm, more than 40, in the different Drosophila species, from 40 to 60, and in the mosquitoes, they are from 70 to more than 100, depending on the species, which makes them attractive for pest control, actually. So, however, there is a little correlation with phylogeny or with ecological niche, which means that animals sharing the same lifestyle do not share similar proteins or a similar number of proteins, actually. Odorant binding proteins usually have 6 cysteines, but some members of this family have only 4, and these are called -C, and while others have 8 cysteines, and these are called +C. The invertebrate OBPs are mainly located in the antennae of insects, and this prompt for role in chemoreception by binding odorants and transporting odorants in the lymph, and acting as a scavenger of odorants. Actually, these proteins can bind a huge variety of molecules, from semiochemicals to nutrients, hormones, and also toxic molecules. Sorry. Okay. However, invertebrate OBPs are not only involved in chemosensation, they are also found in non-sensory organs. For example, they are found in secretory glands, where they participate in the management of pheromones, because they can participate in the storage of this molecule by binding them, and in their slow delivery, and can also protect the molecules from enzymatic degradation. Moreover, it has been suggested that by changing the expression of this protein, it is also possible to change the smell, let's say, of the blend, so the composition of the blend, because different proteins bind different odorants with different affinities, so that by modulating the expression of protein, it is easier to modify the blend than by modulating the synthesis of a small molecule, which is intrinsically less, a more difficult task, actually. Then OBPs are found also in reproductive organs. For example, they participate in egg formation and in sperm composition, and have a role in embryo development and in adult limb regeneration. They can be found also in the venom of some wasp species, and in the buccal apparatus of different insects, for example, in the mosquito, where they can act as anti-inflammatory agents or participate in nutrition. And some OBPs have been found also to participate in the vision process and can confer resistance to insecticides. So actually, they are termed odorant binding protein, but their role, their biological role, well extends beyond the odorant binding. On the other hand, I've mentioned that also vertebrates do have odorant binding proteins, which are completely different. Odorant binding proteins of vertebrates belong to the lipocalin superfamily, which is a huge superfamily of proteins characterized by the ability to transport small ligands, from steroids to retinol to lipids and to odorants as well. Lipocalins are expressed in gram-negative bacteria, plants, invertebrates, and also in vertebrates, and share almost nothing about the sequence, given that the only common motif of these proteins is an amino acid flanked by a glycine and a tryptophan. However, they share the same structure, which is made up of an 8-stranded antiparallel beta barrel connected by loops and an alpha helix that you see here, depicted here. Lipocalins are involved in immune response, in prostaglandin synthesis, in binding different molecules, and can act as pheromones, as hormones, so a huge variety of function for this large family of proteins. Some lipocalins are secreted, some others are not, and some of them have a double binding domain. One inside the protein, in the core of the protein for a ligand, and another one for a receptor. Here you can appreciate how similar is the beta barrel structure of very many different lipocalins. Some lipocalins, not all lipocalins, are listed here in this huge part of the slide. And you can also appreciate that the loops connecting the beta barrel, the beta sheets, are actually very variable in length, in shape, and so on. Now we concentrate on two members of this large family, the odorant binding protein here and the major urinary proteins. The vertebrate odorant binding proteins are found in the nasal mucus of vertebrates, where they constitute roughly 1% of all soluble proteins. Some of them are present as monomers, some as dimers, and so on. All of them have a KD in the micromolar range, which means that they bind the ligands with a rather weak affinity, and this is an important feature because they can bind their ligand but also release their ligand, and this is a property that can be exploited for application. Vertebrates, OBPs, have different functions. They can act as carrier or as a scavenger, and can also be implicated in the clearance of toxic substances. Actually, they can be seen as rather non-specific carriers of small odorants, of small ligands. The last family of proteins that I'm presenting here is the major urinary proteins, who are produced mainly by some rodents like mice and rats, and are coded by a family of genes which is located on chromosome 4 in mice and chromosome 5 in rats. These proteins are synthesized in the liver, released in the bloodstream, and then are excreted by the kidney in urine. However, they are also expressed in exocrine glands like lacrimal glands, salivary glands, submaxillary, mammary, and preputial glands. MUP share roughly 30 positive amino acids homologous to OBP, and they share the same structure, of course, with a pocket with a highly apolar lining. Usually they have an average affinity of 10 to minus 4, 10 to minus 5 molar, with a Km of 0.2 micromolar for the standard ligand, which is IBMP 2-isobutyl-3-methoxypyrazine, which is the smell of the green bell pepper. However, MUP are released in the environment with some ligands bound, and these ligands are the odors which confer the bad smell to male mouse urine. Mainly they are one molecule called thiazolene and the other one is brevicomine. These substances have been described as pheromones for mice, but also the protein backbone can act as pheromone and signal the presence of a mouse. So they are involved in pheromonal communication. So actually all these proteins are very diverse, but have some interesting properties for biotech exploitation. First, they have an hydrophobic binding sites for an hydrophobic ligand, which can be customized quite easily through site-specific mutagenesis. They also have a simple structure with almost no post-translational modification, and they have also a small size, which makes them very easy and cheap to express, let's say, in bacteria. Another feature which is very, very interesting for a biotechnological exploitation is the fact that all these proteins are very, very resistant. They are resistant to temperature, to organic solvents, and also to proteolytic agents. And this is actually very interesting, and this is why we have used some of these proteins to create sensors to replace the sniffer dogs, or as you can see here, the giant pouched rat, for example, to detect explosive or drugs. And this is actually what we did by using proteins from both vertebrates and invertebrates, not actually the dog and the fly, with a biomimetic approach that will be best described by my colleague Krishna Persaud. So I'm pleased to leave the stage to Krishna to explain something more about this.
Thank you, Carla. I'm just going to share my screen now. So Carla's given a very good introduction to the proteins themselves. And the problem now is really how to translate the proteins to the chemical sensors. And I'd like to take you through several steps that we've taken along this pathway today. So we start off with the kind of concept of electronic noses, which is based on the kind of biological system where, in fact, our noses consist of many, many millions of receptors, probably 400 types in human nose, where we have broad but overlapping specificities in terms of the sensors themselves. So if you look at this diagram on the screen where I'm pointing, you can see that we have like a cross-selectivity pattern where we have, say, 3 sensors here that might be responding to a particular ligand or odorant. But this receptor also has some overlap in specificity to this receptor and also to this one. And in fact, when you start putting many receptors together, you can actually extract a huge amount of information which allows us to use a few receptors or a few receptor types, which are very poor in selectivity, and extract a lot of information, which allows us to identify an odor and also to decide whether we like it or not. So when we now look at a kind of a diagram, an engineering diagram of the nose itself, here we have different types of receptors in this layer. And then these are then interconnected to the olfactory bulb, where we have little processing elements called glomeruli. And the signals then are processed in this area and then go on to the cortex, where we make a lot of decisions regarding the chemical stimuli that we're receiving. We can divide this up in kind of engineering terms, in terms of the receptor neuron convergence. We have lots of inputs coming into this processing area. We have some kind of network of neurons here, which are extracting patterns or features from these huge numbers of inputs from the neurons. And we have methods of, for example, focusing on an odor of interest against a background, and also to decide then what to do with the interesting odors that we're actually sensing at the moment. So there's a lot of processing that goes on in the higher areas of the brain, which allows you to take action as a result of the chemical stimuli and decide what to do, whether you like it or you don't like it, whether it's so disgusting that you want to run away from it or not. So we can take advantage of some of those concepts in terms of translating into so-called bioelectronic noses, where we can use the sensors from noses themselves as part of an engineering plan to create artificial systems. So this is from a paper, a review paper published by Dung in Census 2018, which describes some concepts where people have been taking receptor neurons from the nose, growing them in tissue culture, immobilizing them onto a set of electrodes, and then making electrical measurements directly from those cells, where you can then extract information in terms of the sensitivity, selectivity towards different kinds of chemicals that are being sensed. So you can take the human nose, extract cells or proteins from the human nose, from the receptors, translate them into some interface electronics, and extract signals, which allows us to discriminate between one type of smell and another kind of smell. Technology has been moving on, and so for example, magical material of graphene allows us to do also many interesting things in immobilizing proteins. And here is an example of a graphene-based bioelectronic nose, where you have proteins taken from the olfactory neurons, in human, in fact, immobilized onto a surface. And you can then make this into a kind of transistor, where you can present an odor dissolved in an aqueous solution to this transistor and get a signal which is now dependent on the odor that's being presented. And it can actually detect femtomolar or below concentrations of particular smell. Apart from olfactory receptor neurons, which are membrane-bound proteins and which are quite difficult to handle, odorant binding proteins, on the other hand, are actually soluble proteins, as Carla illustrated. And they actually have a binding site that is quite flexible in terms of the number of and types of odors that can actually bind. So in fact, many researchers around the world have been trying to use both olfactory receptors as well as odor binding proteins to make bioelectronic noses. And this is one concept from another laboratory where they've taken the protein, immobilized them onto an array of electrodes here, and which allow them to measure what's called bioimpedance measurements, where if the protein changes its shape when the molecule binds to the protein, then you can see a change in impedance, capacitance, or other electrical characteristics that can be measured. So just to remind you, the insect protein has a kind of a springy alpha helical shape. You see these spirals, makes it actually a very flexible structure. The vertebrate protein is quite different in shape and form. It contains what's called beta sheets and a much more planar structure. And the binding site is actually in between these, what you can think of as ribs in the protein. So we can actually play with both of these types of protein to actually make sensors. But to do that, we need to understand a bit more about the binding sites of the proteins. Luckily for us, X-ray structures are now available for a huge number of odorant binding proteins. And using molecular modeling techniques, we can actually start to explore the complex ligand interactions with the binding sites of proteins. Just to remind you, when you think of a protein binding to a small chemical like an odor, it's quite a complex system because you got a protein, a ligand, and a solvent system because the protein is in an aqueous environment. And you have the interactions and the driving forces that allow the ligand to bind to the protein is driven by more as a thermodynamic equilibrium, which you can measure as a Gibbs free energy. Now, molecular modeling proteins are not so good, but they do allow us to get a kind of picture of the protein structure and where ligands bind to the protein. So here we can actually measure, for example, how long a small molecule will interact with certain surfaces within the protein, and by that deduce which is the binding site and which are the important amino acids in the protein that are involved in interacting with the ligands. So here we've taken two proteins from the Anopheles mosquito, and we've been able then to define the binding pocket within that protein that is illustrated here in violet. So this is OBP1, and here we have actually 31 amino acids that we've identified that are very important in having direct contact with the odor that's actually binding to this particular mosquito protein. Mosquitoes actually have a lot of OBPs, and there's another one here that we illustrate, which is OBP47. And this protein has a much different binding site in shape. It's got actually 60 amino acids that line this pocket, and 39 of these residues, these amino acids, are very hydrophobic. And these actually then define the types of ligands, the type of odors that will actually bind to the protein. So really the size, the shape, the charge of the interactions of the ligand and the protein are very important in defining the binding sites. So we've done a lot of work in kind of trying to understand how these OBPs actually interact with small molecules. So we can then think about actually modifying those proteins to bind non-native ligands. So we can actually create mutations of those proteins. So what we've done is to examine these proteins and identify which amino acids can be substituted with other amino acids without compromising the binding pocket on the integrity of the protein. Because one of the problems is that if you kind of make a random substitution of an amino acid within a protein, the whole tertiary structure, the whole three-dimensional structure of the protein just collapses. So we can do a lot of analysis, calculations, which allows us to identify where we can substitute amino acids and where we can actually create new proteins using the skeleton of an odorant binding protein, but modifying those proteins to bind completely different ligands from what they would bind normally. To illustrate, we made a kind of designer protein for cocaine. So we took that OBP1 from the mosquito protein, and when you look at the molecular model of the protein, we find that this is the binding site of the protein, but the cocaine molecule is just too big. It just can't enter into the mouth of that binding pocket. However, by actually making one substitution, just one amino acid substitution in this protein, we can actually change that protein so that the cocaine molecule now enters very happily into the binding pocket of the OBP. So this now gives us the basis of a specific detector for cocaine, which doesn't actually exist in nature. We can do the same for other molecules. So for example, this is binding a molecule of trinitroptoluene, an explosive substance, to an OBP. And here we can actually show that this particular molecule can actually bind directly into the binding pocket. So having defined theoretically that we can actually substitute amino acids, we can make, we know the amino acid sequences of these proteins. What we can do is to make these proteins in quantity in the laboratory. So what we do is extract the DNA, we actually synthesize the DNA of the OBPs. We clone them into something that looks like a small virus. It's called a plasmid. We introduce that small entity into a bacterium, where we now persuade the DNA of the protein synthesis mechanism within the bacterium to actually produce our odorant binding proteins in quantity. So we can actually then express the proteins, and this is an electrophoretic gel which shows a number of proteins that are being secreted by this bacterium, of which you see a huge band here, which corresponds to one of the OBPs of interest that we've expressed. So we can then purify that through normal chromatographic steps and so on to produce a pure protein that you see again on a band on an electrophoretic gel, which allows us then to characterize the protein and do other things with it. So having got the protein in quantity, I mean, in fact, we can now make hundreds of milligrams to grams of protein this way. We then have now the means to use these proteins in artificial systems. So one of the problems we have is really how to keep these things stable, how to expose the binding sites to the external atmosphere. And we've devised methods where you can take a molecule here called thioctonic acid. These sulfur atoms have a very strong affinity to gold. So if we have a gold surface, we can just treat that with thioctonic acid, and this forms what we call a self-assembled monolayer. So they kind of have these, they kind of assemble into this kind of very regular structure here, which allows us then to immobilize our protein at the end covalently. So we have a chemical reaction which allows us to immobilize the protein at the end of that chain. This now gives us a surface which we can then use as part of a transducer. And this can be a quartz crystal microbalance, it can be a surface acoustic wave transducer, it can be an electrode, which allows us to measure impedance, or it could be many other kinds of techniques. So I'm going to now hand over to Emmanuel to continue the story.
Okay, thank you very much, Krishna. Can you all see my screen now?
Yes.
Yes. Okay, so first of all, I'd like to thank Krishna and all the organizers of this webinar for giving me the opportunity to present this work. Krishna was just talking about golds. I'm now going to talk about diamonds, and I guess the connection between diamonds and olfaction may not be very obvious. So I will start this presentation with a few words about synthetic diamonds. And synthetic diamond basically is the same diamond as we find in nature. This is the crystalline form of carbon. This is sp3 hybridized carbon. And the only difference is that this diamond is grown in the lab. And diamond can be grown in the lab using these big machines. It only grows on diamonds, so we need to start with a diamond seed. So this is an example of this is a single crystal diamond plate. We put it in the reactor and in a hydrogen plasma in the presence of methane. Methane is a source of carbon to grow our diamond, and after a few hours of growth, this is what we get, a thick diamond film. If we polish it, we can get some very nice diamond crystals like these. They're beautiful, but they're not very useful for making sensors. So an alternative is to deposit some diamond nanoparticles over, for instance, a silicon substrate. So they look like these. These little dots here are nano diamonds. They are typically between 5 and 20 nanometers in size. And as we grow these nanoparticles in the same reactor, in the same conditions, this is what we get at the end, a thin film of polycrystalline diamonds. And this is a lot better for us for sensing applications. So what can we do with these diamonds? We can do some microstructuration. This is mostly done in the clean room. So this is, for instance, a 4 inches wafer, silicon wafer, that is covered with diamonds. And you can see a lot of structures here. So the dark areas on the wafer are diamonds. Then we have gold contacts to get some electrical contacts. And we can make in this way a lot of different sensing technologies, microelectrodes, resonant devices, micro hot plates, and so on for sensing applications. We can also nanostructure these diamonds. You have an example here, for instance, of diamond that was grown on carbon nanotubes. So again, these are nano size, they're very small. And by doing this on the surface of a sensor, we can actually increase the surface area of our sensors, and therefore the number of active sites that will respond to an analyte, and therefore we can increase the sensitivity of our sensor. We have some technologies also to report these diamonds onto flexible substrates for some sensing applications where we want to go in a pipe or things like these. And last but not least, we can do surface functionalizations. And I just mentioned before that diamond is carbon, and we have here all the organic chemistry available to do some surface modifications and to immobilize proteins, for instance. I will come back to this a little bit later. So this is a summary of what a chemical sensor is. Basically, this is we have a transducer and a sensitive coating. The transducer here is translating the chemical signal into a useful electrical signal. So there is a wide range of transducers that can be found in the literature that have been developed across the world, some electrodes, some gravimetric sensors, and so on. And in terms of sensitive coatings also, there is a wide range of coatings that can be used, polymers, carbon nanotubes, and so on. So what we're actually doing in this lab is we're trying to take advantage of all the nice properties of diamonds, of this synthetic diamond, to get some high performance transducers and combine those with biological materials that are actually involved in nature in olfactions. And this includes the odorant binding proteins and the major urinary proteins that have been described by Krishna and Carla, and also olfactory receptors. So why are we interested in diamond for chemical or biochemical sensing? I will be very brief here, but basically if we look at the physical properties of diamond, diamond is probably the best materials in all the areas of physics. It's the most transparent material. This is a material that has the highest thermal conductivity, that propagates the acoustic waves fastest. It has outstanding mechanical properties. If you look at the Young's modulus here of diamonds compared to silicone, for instance, it's about 7 times better. So that means we can use all these properties to enhance the sensitivity and the performances of our chemical sensors and especially our transducers. Diamond is also a powerful substrate for protein-based sensors. And there are a few reasons for this. The first one is that when doped with boron, this diamond becomes a semiconductor. But by contrast with silicone, for instance, it has no native oxide layer on its surface because the oxidized form of carbon is CO2, which is volatile. So we have a semiconductor here where we can use the surface directly to interface it with any biological material. So you have an example here, for instance, where we're using the hydrogen-terminated surface of diamond, which is a p-type semiconductor. And we are monitoring in this way some selectivities on the surface of this diamond directly using a field effect transistor configuration. Diamond is also a great electrode. It's the electrode that has the widest potential window in water. It has a low capacitive behavior, so it's a fantastic electrode for electrochemists. We can immobilize proteins and do a lot of electrochemistry on them. And it's also a biocompatible material. In fact, besides all this work that we are doing on chemical sensors, we are also working on diamonds for biomedical implants. And in this context, we did a lot of biocompatibility studies, and we demonstrated that this diamond is biocompatible. So it likes, cells are very happy to grow on diamond and so on. And lastly, as I mentioned before, diamond is carbon, so we can use a lot of different chemical routes to immobilize some proteins and some linkers on the surface of these diamonds. Krishna has been talking about the use of these linkers on the surface between the receptor and the sensor surface. One of the great advantage here of diamond is that because it is carbon and we can do some carbon chemistry on it, we can immobilize linkers which are covalently bonded to the diamond surface using carbon-carbon bond, which are, as you all know, one of the strongest chemical bond in chemistry. So we can make very stable attachment of proteins on the surface. So this is a typical example of how we can immobilize a protein on the surface of diamonds. And so if we take, for instance, a native diamond surface that comes out of the reactor, it's hydrogen terminated on the surface. If we expose it to an aliphatic amine, which contains on the other side here a receptor, and if we put this system in some proper pH conditions, we are actually protonating the amine group here. We're forming a carbanion on the surface, and through a nucleophilic substitutions, we end up with this carbon-carbon bond on the surface. So this is a single-step attachment. It's covalently bonded using carbon bond on the surface, so it's highly stable. So an example of applications is this nitriloacetic group here, NTA, that I'm sure many of you know because it's used quite commonly to purify proteins. And so this group here is terminated with an amine. We can attach it in this way to the diamond surface. And then we can expose it to some metal ions like nickel. It will form this complex, which will then interact with ethylene tags that are attached to the proteins. And the beauty about these tags is that they can actually be located on the protein. That means we can actually control the orientation of the protein on the surface. And as Krishna and Carla described before, OBPs and MUPs, for instance, they have a binding site which is localized on the protein, and it is crucial that this binding site is actually facing up, so it can actually bind with the ligand. So this is a very efficient way to immobilize this protein on diamond surfaces. We can also use some electrochemical approaches if we have a boron-doped diamond. So basically, the first steps are some electrochemical deposition of the binding of the linker, and then through some peptidic attachment, we can proceed to a very similar way. One of the advantages of this method here is that we can actually use some electrochemical impedance spectroscopy to monitor the attachment of the protein over the surface. So if we're looking at this diagram here, this is a Nyquist plot. Basically, we are looking at the transfer resistance of the electrode. This is the ability of the electrode or the ability of an electron to pass the surface, the interface between the electrodes and the liquids. And this is seen here as the radius of the semicircle here on the Nyquist plot. So you see that on the bare diamond, this radius is very small. The transfer resistance is small because there is no organic material on the surface. But if we add some organic materials like NTA on the surface and then OBPs, for instance, this transfer resistance will increase because these organic materials will generate a barrier that will prevent electrons to go across the electrode material. So this is a good way to characterize the immobilization of the proteins, but we can actually use exactly the same principle to make a biosensor. So an example of this is this surface here, which has been functionalized with membrane fragments from yeast that contains an olfactory receptor. In this case, this is the human OR 1740. And what we do again is we monitor the impedance of the electrode as we are exposing this electrode to different ligands. So this receptor is well known to have a high affinity with Elional, for instance. So as we expose this sensor to different concentration of Elional, we have some change in the conformation of the protein that will affect the transfer resistance of our electrode. And you can see that if the concentration increases, we have an increase in the transfer resistance of our electrode. So we do have a sensing signal. And you can see that we can get down to the picomolar range. So we are extremely sensitive to our ligands here. So we can apply this to different types of olfactory receptors. And if you look here are some examples of responses for two different receptors. This is from OR7D4 from the chimpanzee, and this is the human receptor. For the same ligands, they have a different behavior. So we are moving towards the principle of what Krishna was explaining about the bioelectronic nose. We can create an array where with a combinational approach where each sensor will have a broad selectivity to different analytes. So this is a summary of the same results. What is very interesting about these olfactory receptors is that sometimes they can be quite selective. For instance, OR7D4 has a very high affinity with vanillin, whereas the OR1740 has almost no response to vanillin. Yeah. So basically, the sensor works quite well. If we look at the response and we compare to affinity constants that are reported in the literature, there is a very good agreement. So we have a beautiful sensor, a different approach, which is quite similar. We're using a different transducer here. We are using a diamond micro cantilever. So this is a diamond beam that was grown on silicon. We removed the silicon underneath. The size here is about a few hundred microns in length, a few microns in thickness. And as we immobilize some olfactory receptors on the surface, and these receptors are interacting with ligands, they are actually increasing the mass load of this cantilever. And if we bring this sensor to resonance, then the resonance frequency of the device will be affected by mass loading on the surface by interaction with ligands. So we are getting similar results. Again, some very high, nice selectivity, for instance, with the mouse olfactory receptor M71, which is known to have a high affinity with acetophenone and not so much with 2-octanone. And indeed, we can see that our sensor is very selective to acetophenone. So this is very interesting, but we're working in the liquid phase. And when we're interested in an electronic nose for olfactions, we are mostly measuring in the gas phase. So these proteins are membrane-based and they're not very happy to be in a dry phase. And also we have very few data about the stability of these proteins. So all the results that I have been showing here have been measured on the same day as the sensors were prepared. So we move to other types of sensors that are actually using ligand binding proteins, which are OBPs and MUPs that were described by Krishna and Carla. And as Krishna and Carla said, these proteins are very stable, and this is why we are so much interested in them for artificial olfaction applications. So this is some work that was done in the framework of a European project that I will come back on later on. So we collaborated with Krishna and Carla's team, and they did a fantastic work in screening and expressing a broad range of OBPs and MUPs, and look at the affinity of these proteins towards different chemicals. So we were mostly for these applications interested in chemicals that are related to explosives or narcotics for security applications. So as Krishna described, some of these proteins were mutated to be specifically specific, for instance, to cocaine. So they did this great work, and meanwhile we developed some diamond-based transducers onto which to immobilize these proteins. So we were interested here in surface acoustic wave sensors. The principle of this basically is it's a piece of piezoelectric material onto which we deposit some IDT electrodes. And as we apply a potential to these electrodes, we generate an acoustic wave that will propagate across the surface of the device. And this device can be brought to resonance. So it's resonating. If we deposit a sensitive coating in between these two electrodes, and if we have some interaction of this coating with analyte, this will disturb the propagation of the acoustic wave, and this is our measuring signal. So what is very interesting about these sensors is that if you look at the propagation of the wave here, this is a cross-section of the device, you see that all of the acoustic energy is very much confined to the surface. That means that these sensors are very, very sensitive to what is happening at the surface of the device. This is very good because we can make very sensitive sensors, but this has a major drawback also because they are very sensitive. They are also very sensitive to the sensitive coating that we put on the surface. And we're talking here about thicknesses that are in the range of 10 to 100 nanometers. And if we are trying to deposit polymers, for instance, it would be very difficult to make reproducible sensors just because the thickness of the sensitive coatings will vary. So this is why we developed this coating based on nanodiamond. So we are depositing nanodiamond particles across the surface of our surface acoustic wave sensors. So this, what you can see here, is the gold electrodes of the IDT electrodes, and they have been conformably coated with these diamond nanoparticles. So what we get at the end is a carbon surface, which is highly stable because it's diamonds. It's nanoparticles, so we have a very high surface area, so we can make a very sensitive sensor. And we can also use this carbon surface to immobilize protein on the surface of these sensors. So this is what we did. I will not come back to this because I already explained how this works in the previous slide. We use the same approach to immobilize some odorant binding proteins and MUPs on the surface of these transducers. And so we're looking at the resonance frequency of our device. This is a resonance peak. As we are adding the different functions on the sensors, we have a shift towards the lowest frequency because we are adding some mass loading on the surface. But after the last step of immobilization of the proteins, we can see that we still have a very nice signal. So we can start now to expose our sensors to different odors, different VOCs. And this is the typical responses that we're getting. Here we're looking at the response for 4 different sensors. 3 of them are functionalized with MUPS, 1 with OBP. And you can see that they're very sensitive. Here we are measuring 70 ppbs of 2,4-DNT. This is an analog, structure to explosive TNT. So we are very sensitive, and you can see also that the sensitivity varies from sensor to sensor because the affinity of the different proteins is different towards DNT. We also look at the stability of the sensors, and so here I'm just showing an example of response. This is a sensor that was functionalized with wild type pig OBP, and we expose it to 10 ppm IBMP on the first day of preparation, and then after 1 month and 3 months, and you can see that the response is very much the same after 3 months of storage in some fairly standard conditions. So we built a sensor array with using this principle, and what we did is we look at the sensitivity of the different sensors with different proteins on the surface to different analytes. So here I'm showing an example of an array which contains only different maps on different sensors. And what we did is some calibration curve, basically in the liquid phase, in the gas phase. Sorry. So this in this example, I'm showing the response curves for nitrotoluene, and you can see that each protein is indeed giving a different sensitivity to different to the same analyte. So what we did in parallel is we actually looked at the affinity of these proteins in the liquid phase, using a competitive binding assay, using displacement of a fluorescent probe. This gives us the affinity constant of our protein in the liquid phase. And we compare the affinity constant in the liquid phase with the sensitivity in the gas phase. And for most proteins, what we found is that we had indeed a fairly good correlation between the sensitivity in the gas phase and in the liquid phase, which is suggesting that our proteins are actually behaving in a very similar way, both in the dry phase and in the liquid phase. I have to say that these proteins had to be kept hydrated. This is working perfectly fine in normal air, in the atmosphere. If we store the proteins in the fridge, they get dehydrated, and then we have to hydrate them again before they work. So it shows the importance of keeping the proteins hydrated. But apart from that, they are very stable in air, and they have a very similar response to the liquid phase. So we build up a sensor array, and I'm moving now to some applications. So we've been talking a lot about narcotics and explosives. We were actually performing all this work in the framework of 2 projects, the Sniffer project and the Seaboard project, and they are both related to security applications. The Sniffer project was focusing on the detection of explosives and narcotics in airports. So these 2 families of compounds, explosives and narcotics, they are mostly chemicals that have very low vapor pressures. So if we want to try to detect them, they are present in the form of particles. This is why when we go to the airport and you go to security check, they will try to collect particles of you on a filter, and they will analyze it with an IMS machine. So we use a similar approach, and to collect these particles from different luggages, we had partners that developed different sampling tools, some kind of aspirating systems and so on, to extract these particles from the luggage and on the filter, and this filter was then placed on thermal desorption unit to vaporize these chemicals into the gas phase. And here we are looking at a typical response for 12 nanograms of TNT, which is of course an explosive compound, when it is exposed to our sensor array, which contains a range of MUPS and OBPs. And you can see that we have a high sensitivity. 12 nanograms is typically what we're trying to achieve in the real applications. So we are meeting this target. And you can see again that each sensor has a different sensitivity to the different to the same TNT analyte in this case. We also work and develop further this technology in the framework of the Seaboard projects. We are the the objectives are similar here, but we were focusing on freight containers, looking also at some explosives mostly, and also some illegal goods that may be hidden in the containers. So we were also trying to collect particles. For this, we had to inject some pressurized air to lift up the particles, and then they were collected in a filter, and then again thermally dissolved in this bench here that could be taken to the field. So I'm just showing some results from the sniffer project. So this is some typical results that we obtain with our sensor array, which again contain a selection of MUPs and OBPs that were carefully selected because of their high affinity to these compounds. We're looking here at the steady state response of each sensor to different chemicals, cocaine, cannabinol, heroin, and so on. Here some explosives. You can see that each sensor has a very different affinity to these chemicals, and mostly they're showing very different patterns. And these patterns can actually be trained using artificial neural networks by the machine to learn and recognize later in the field these chemicals. And this is what we did. So we took our device to the field, we benchmarked it against an IMS machine, and so the device was trained in the lab first. And here I have summarized a few results from the field. So unfortunately in the airport we were not allowed to use narcotics for security reasons, so we only work with explosives. But you can see with all the explosives that we hide in different bags, we had a positive response from our electronic nose or our bioelectronic nose. And these are some typical outputs from the artificial neural network. And you can see also that we had a fairly good identification of the different explosives in the bags. So I will finish these presentations with this. Thank you very much for your attention. And I guess we are very happy to answer your questions.
Thank you very much indeed, Emmanuel. Thank all three of you for a very nice presentation, beautifully put together and following on. And I just hope you're funding it by making diamonds from methane in your microwaves. I will check now for questions. I just wanted to ask why you didn't think that the things were in a liquid phase in the nose, because the nose is always wet with mucus.
Yes, this is correct. My comment was more related to the fact that we are sampling most of the time VOCs in the gas phase. But okay, we have thought about this, in fact, of having a bioelectronic nose where the sensitive coating is actually in a liquid gel, in a gel or in a liquid phase somehow. And so we are considering this indeed, because this is how nature works. Yeah. But my comment was more related to the fact that in the applications we are extracting analytes from the air. So we need a device that is able to work in the air, or at least to trap chemicals to then a liquid phase and then to the sensor. But at the beginning, it all starts from the air, yeah, in most applications anyway.
Okay, thank you. Now, if anyone wants to ask a question, there's a thing called chat at the bottom. I'm just opening it up to see if anybody has sent a question. Not yet. If anyone would like to ask one, you can just put up your hand, either real like that, or there is a virtual hand you can raise at the corner and actually ask a question. Do you have anybody with anything to ask about? Well, can I ask something else? In my hospital, they are going to be trying dogs to detect COVID-19. And I wonder if there's any possibility of using your system for this. And I don't know what the smell of COVID-19 is, but presumably it does, in the infected patient, have a distinct odor which dogs might be able to detect. Do you have any thoughts on that?
Krishna, you want to comment on this? I know there are many applications for cancer research where indeed the smell of the patient is not necessarily related directly to the disease, but it's a side effect. Like, you know, in the COVID case, of course, we are not detecting a virus, but some odors that are related to the conditions of the patients. So there are a lot of quite promising studies. And in fact, I was in contact some time ago with a clinician from emergency unit, and he was telling me that when he entered the room of a patient, he can tell if there is something wrong. And so he was asking, he said, oh, can we have an electronic nose? Because I can smell with my nose. I know sometimes that something is wrong just by smelling. When I get in the room, I know that the smell is not normal, you know.
Yeah. Yes, in fact, any kind of infection that you have, whether it's a viral infection or bacterial infection, will cause change in metabolism in the human body. And as a result, the secretions and your breath, your urine and sweat will all change in composition. And you can actually, as reflected in change of smell. So it's quite possible that with COVID, something will happen that will allow you to detect a change in condition. I'm not sure whether it will necessarily detect COVID specifically, but I think it will certainly be able to detect a change in the condition of the patient. There is a message in the chat room.
Yes, I can just see it. Can you, from Johannes Bintker and also from Sachiko Koyama, can you comment on the thermal and humidity stability of the sensors, please? So
maybe this Krishna or Carla can comment on the stability of the proteins first, because this the sensors that I have been describing at the end of this presentation with OBP and MEPS are relying on the stability of the proteins. So we have the diamond is very stable. That's one thing. But the weak point most of the time is the sensitive coating, and here we are working with very stable proteins. So I already made a small comment about humidity. You know, we noticed that when we store the sensors in the fridge, for instance, they get dehydrated and they don't work so well. But if we just dip them in water for a while and then dry them in air, then they work again. I think Krishna and Carla, I think you have more experience than I have on the stability, but so maybe you can comment on it.
I can comment on the stability of the natural major urinary protein. We can see some changes in conformation about around 70 degrees Celsius. So it means it denatures, it changes its conformation up to from 70 degrees onward. And about thermal stability, this is about thermal stability. I do not have direct measurements about stability to UV, let's say in the sunlight, but by sure we have measurement taken from the environment with differential release of volatiles, and we went on measuring the release of volatiles for at least 48 hours by sure, and then And about the proteolytic sensitivity, there is a differential sensitivity to this differential proteases. In some cases, let's say, the major urinary proteins are not very sensitive to trypsin, for example. They are rather stable. And while with other tryptic digestion with pronase, they are more sensitive. So, but they are usually more resistant than other proteins.
That's right. All proteins will actually unfold when heated. But one unique aspect with OBPs is that you can heat them up, they actually unfold, they change conformation, but when they are cooled down again, they actually refold back to the original conformation. So they actually work as normal when they reach back to normal temperatures.
Gosh. Can I ask you, Krishna, when you want to alter the binding pocket in order to make something fit in, do you do it theoretically first and then make that molecule, or do you just try a whole range of things and see what works?
No, in fact, we have to do it theoretically because really there's so many possibilities, so many combinations that's impossible to do that experimentally. So what we do is do the calculations first, decide which are the most likely propositions, and then we make those proteins experimentally.
Thank you. Okay. Using your, this is from John to everyone, using your detector in practical terms, what is the threshold amount of TNT that a passenger could bring through an airport without being detected?
The smallest amount, well, I think it's probably the best detector is the dog today. So the smallest amount is the threshold that the dog would be able to detect. Talking about instruments, I was mentioning about the IMS today. The typical limit of detection for these devices in the order of 1 nanogram. And so I'm not sure, I don't know if Krishna, you have some ideas about the threshold for the dog, but the dog is you know, is the gold standard. It's really good. So I don't know if he's able to detect 1 nanogram, but this would be typically the order of magnitude of what we're trying to detect. 1 nanogram, it's not very much.
No, it isn't at all.
Good, good. That is reassuring. We have a comment from Sachiko Koyama about dogs. She saw a news item that they can precisely detect COVID-19 odors, but they can also get infected. And she doesn't know whether the infection reduces their ability to smell, as it can do in humans. So that's, one have to use dogs that have been vaccinated or have recovered from the infection, I guess. Okay. Well, if we don't have any more questions, anybody else want to ask any further questions? No? But it remains to me to thank you all three, Krishna, Carla, and Emmanuel, very much indeed. That was really an enlightening talk. One second. We have a final question from Johannes. For the fabrication of the sensor array, how do you immobilize multiple OBPs on the array?
This is a very interesting question. It depends, of course, on the technology that is used for transducers. The surface acoustic wave sensors that have been describing, they are actually single sensors. They are very small, but they can be manipulated individually. So what we normally do is we make batches of sensors which are immobilized with the same proteins. So typically we could make like 10 sensors with the same protein, and then 10 other sensors with a different protein. And in that way, we can build 10 sensor arrays very quickly, you see. It becomes an issue indeed if all the sensors in the array are already mounted on the same chip. And but there are different approaches for this. I was talking about electrochemical immobilizations. If we immobilize the linker one by one, then the sensors which have no linkers, of course, they cannot have proteins immobilized. So we can do it in a sequence like this. I'm not sure this is very reliable because we may have some proteins from the neighboring sensors that could also fix on different sensors. But the way we do it today is we functionalize sensors individually or by batches with the same protein, because we have this chance that sensors comes as an individual chip in our case.
Thank you. Thank you all very much for your excellent talks. Thank you, Krishna, for organizing this, and thank you all for attending. It's been another great UKSN meeting, and I think we're all, oh, Sachiko has, oh, it's clapping. Thank you very much indeed.
Thank you.
Bye.
Thank you for the invitation.
Goodbye. Bye.
Bye.
Thank you.
Bye.